Hello, ECOL340. How are you? Today, what we're going to do is we're going to take a little bit of a deeper dive in terms of understanding the evolution of photosynthetic diversity in the land plants. And in particular, what I would like to do today is kind of highlight some important aspects of photorespiration. In particular, why then photorespiration is actually an important constraint in terms of influencing not only the evolution of photosynthetic organisms on the planet, but also the evolution of diverse photosynthetic pathways. And it turns out photorespiration has been important in terms of the evolution of various photosynthetic pathways. So in the past, in your past biology classes, you've probably been introduced to kind of the interesting kind of details of photosynthesis, and you've learned about C3 photosynthesis. But it turns out that photosynthetic pathways across the land plants actually vary quite a bit. And so today we're going to be providing a brief overview then of this important variation. In particular, we're going to overview some different flavors of photosynthesis, not just C3 photosynthesis, but today we're going to be learning a little bit about C4 and CAM photosynthesis. And in particular, we're going to be looking at the important ways to distinguish differences between C3, C4, and CAM photosynthesis in terms of their biochemical and anatomical differences, as well as how they functionally differ when compared with C3 photosynthesis. This whole backdrop then is placed in terms of we want to understand the unique selective pressures leading then to the evolution of C4 and CAM photosynthesis. And we're going to try to kind of piece together some of how we understand the unique selective pressures leading to C4 and CAM photosynthesis by looking at geographically where do we tend to see C4 and CAM photosynthesis in terms of where they tend to be more dominant. So we're going to piece all this together in terms of thinking of variable photosynthetic strategies. Okay? And in particular, kind of when you've been taught kind of in introductory biology, C3 photosynthesis, as it turns out, once we start investigating a little deeper, it's not as simple as you think. There's actually quite a bit of variability in photosynthetic strategies. Okay, so big picture, stepping back. If you remember, in terms of our overall carbon cycle reactions, if we look at the Calvin cycle here, we have this, again, this biochemical water wheel, right? Where we have then within the chloroplast, our light reactions then interacting then with our carbon reactions or our dark reactions, all within the chloroplast. And we have this important kind of enzymatic step where Rubisco, remember this key enzyme, is interacting then with carbon dioxide, then that's diffusing then from the outside atmosphere, interacting then with this other molecule, RUBP, to then form this 3-carbon molecule, 3-phosphoglycerate. Okay. And so this 3-PGA then is this first then molecule. Okay, that's characteristic then of C3 photosynthesis, this 3-carbon initial molecule. Okay, so if you remember back, we talked about the Calvin cycle, these carbon then reactions, and that provides then our framework for today. And so last time I introduced photorespiration. And so we should think of photorespiration as potentially a problem with C3 photosynthesis in certain environments. And in certain environments, we tend to see an increase in photorespiration, mainly in hot, dry conditions. In conditions where stomates are closed, concentrations then of CO2 within the leaf then start to decrease, right? Because CO2 is no longer diffusing in freely from the outside. We get a decrease then in CO2, and because of which the relative concentrations of oxygen compared to CO2 then increase, and because of which Rubisco then starts acting like a different enzyme. Okay? No longer is a carboxylase, it now starts interacting with oxygen. Okay, and so this occurs because there's competition between oxygen and CO2 then for the binding site on the enzyme Rubisco. And so photorespiration increases when the relative concentrations of oxygen and CO2 inside the leaf change so that there's relatively more oxygen compared to CO2. And that tends to happen when temperature increases, okay? And the plants then are drought stressed. So rubisco, remember, is not only a carboxylase, but is also an oxygenase. And in the case when it acts like an oxygenase, the end result is it's going to be catalyzing a reaction between RUBP and now oxygen. And in the end, that leads then to photorespiration, a product phosphoglycolate now, then, and the breakdown of phosphoglycolate leads then to the release of CO2, hence photorespiration. CO2 then being emitted during the day when there's light, photo. And remember that this is a form of respiration then that's outside then of the normal mitochondria. So photorespiration. So like respiration, this process utilizes oxygen and produces CO2, but unlike respiration, it ceases in the dark. And the substrate is RUBP, right? And rubisco then, remember, has a much greater affinity for CO2 than oxygen. All right. So under natural conditions, remember, when concentrations of oxygen are around 21%, concentrations then of CO2 about 0.03%, C3 plants immediately lose 20% then of their photosynthetically acquired CO2 in the form of photorespiratory then loss. Okay. And so if you remember, I gave you several different examples from last class in terms of when we tend to see photorespiration then occurring. And in particular, I highlighted a few different experiments. So here's an experiment where we get changes in light intensity with increases in photosynthesis. And if we then grow plants in either a low oxygen environment or a high oxygen environment, we immediately see a difference then in your photosynthetic response then to light. This difference is totally due to photorespiration. So notice once we start decreasing the relative concentration then of oxygen relative then to our normal concentrations of oxygen, photosynthetic rates increase because we're lowering then that relative ratio between oxygen and CO2. Okay? And so C3 plants actually do better in lower oxygen environments. So if you remember our ACI curve, okay? And in the ACI curve, as we increase the internal concentration then of CO2, our photosynthetic rates increases. Remember, this linear part is called the demand function. As we increase CO2, remember, because we have so much RUBP, rubisco then starts to operate faster and faster and faster as we increase the relative amount of CO2. But it gets to a point where we come to this balance point, and ultimately rates of RUBP regeneration are going to be what's limiting photosynthesis. That's the ACI curve, if you remember for last time. But notice this one point right here. This is called, remember, the compensation point. Okay. So this concentration point, the CO2 compensation point, is very important because it turns out that different plants differ in their compensation points. So again, I'm showing you some ACI curves then for 3 different plants, actually 2 different plants. The top curve, the solid curve, is sorghum, okay? It's a crop plant, and it's an important crop plant that's we're going to get to in a little bit. The other plant is wheat, but wheat grown, this is a C3 plant, but wheat grown at 2% oxygen, and wheat grown in an atmosphere with 21% oxygen, normal conditions. Notice what happens to this compensation point. Sorghum has a compensation point that goes to 0. That is, it can perform photosynthesis at really, really low concentrations of CO2. Now, wheat, under normal conditions, notice its compensation point, where photosynthetic rate then goes to 0, is much higher in terms of its intracellular concentration of CO2. But if we grow wheat at a lower oxygen concentration, it can basically perform photosynthesis almost down to very low CO2 levels because we're limiting then photorespiration. Okay, but what's weird with sorghum here, if we grow it at 2% oxygen or at 21% oxygen, there's no change in its relative photosynthetic rate. Now in wheat, which is a C3 photosynthesis, we do see a difference. So why then are some plants very sensitive in their compensation point? Okay. So it turns out that the CO2 compensation point is an important measure for understanding photosynthetic diversity. All right, so let's try to place what we've learned about C3 photosynthesis, okay, into kind of a context of what actually limits C3 photosynthesis. So ask yourself, what environmental conditions does photorespiration appear to become a detriment to the carbon balance of the plant? So we told you at kind of relatively high oxygen concentrations, right, in times of drought, okay, high light availability when stomates then tend to close. Now, given the constraints then of Rubisco and the given environmental conditions where photorespiration tends to be high in kind of really warm, dry conditions, right, how can natural selection improve upon the photosynthetic performance in plants? So immediately, remember, with the invasion of land by the land plants, we start to see in these environments in hot, dry, high light conditions, such as in grasslands, deserts, high alpine environments, right? These are the environments then, especially the hot and dry environments, where C3 photosynthesis starts to show some limitations, okay? So think about plant macroevolution here. Which environments were among the last to be colonized? Okay. Hot, dry environments. Now, why was that? Well, water was limiting, right? But also C3 photosynthesis starts to meet some real challenges within these environments. So since the colonization of land, has natural selection actually shaped the efficiency of Rubisco, right? Maybe in certain plants, such as sorghum, I showed you that example, maybe photosynthetic diversity is reflecting natural selection shaping the efficiency of Rubisco. Well, it turns out that Rubisco hasn't changed, basically, since the origin of the foundations of C3 photosynthesis. But instead, natural selection has come up with several ingenious ways to avoid respiration. And plants do that. Certain plants do that by excluding oxygen from the Calvin cycle. Natural selection has come up with ingenious ways to do this. And so it really wasn't until the last, say, 50 or so years that we've really kind of sketched out the importance of photosynthetic diversity. So there's pathway variability in terms of photosynthesis. So we've learned a lot about C3 photosynthesis, but variation in biochemistry and photosynthesis exists and has important ecological and evolutionary implications, in particular CAM and C4 photosynthesis. And so the biochemistry then of this photosynthetic pathway variation was worked out back in the 60s, actually by Hatch and Slack in 1966. And C4 and CAM photosynthesis are two evolutionary solutions to the problem of maintaining photosynthesis with stomates partially or completely shut on hot, dry days. You can think of these differences in photosynthetic pathways as carbon concentrating adaptations. Adaptations to effectively lower this concentration difference between oxygen and CO2. So these are carbon CO2 concentrating adaptations. Okay, so remember in C3 photosynthesis, CO2 is fixed by Rubisco, and then this 3-PGA, this is a 3-carbon molecule, is the product of initial carboxylation. And in C4 plants, turns out CO2 fixation is no longer done initially by PEP carboxylase. I'm sorry, is no longer done initially by Rubisco, but is instead done initially by PEP carboxylase. So in C4 plants, we have a new enzyme, PEP carboxylase. It's now interacting then with CO2 to then produce what's known as OAA. This is a molecule that's a 4-carbon acid. So instead of a 3-carbon molecule being produced in C4 plants, a 4-carbon molecule is produced, hence 4 in C4. Okay, so this 4-carbon acid then is transported within the leaf where we have a decarboxylation event, and then we have a refixation within the plant by rubisco. So rubisco is still there within C4 plants. It's just hidden within this more complicated biochemistry. In CAM plants, crassulacean acid metabolism, CAM, C-A-M, CO2 fixation is then by PEP carboxylase again to produce a 4-carbon acid, OAA. But the results of this production then is going to end with producing malic acid in the vacuole of the plant during the day, but then we're going to have decarboxylation and fixation by rubisco again at night. Okay, so this is a short overview of the fundamental differences between C3, CAM, and C4 plants. Now, let's go into a little bit more detail. So C4 photosynthesis then results in CO2 being concentrated at the site of carboxylation in order to basically MacGyver your way around the issue of photorespiration. C4 photosynthesis is a spatial specialization of biochemistry. Has to do with how then we're spatially then separating out differences in the biochemistry. Now, what's interesting about C4 plants is that you can tell the spatial specialization of biochemistry by cross-sectioning then the plant, the leaf in particular, and looking on the inside. And you can see these specialized anatomy then that separates out where our normal kind of C3 components of photosynthesis is occurring, but then also this outer layer onion then of C4 photosynthesis. CAM photosynthesis results in CO2 concentration at the site of carboxylation by temporal specialization. So we're no longer separating this more complicated biochemistry in terms of space, but instead we can separate it in time. Ooh, interesting. So these different photosynthetic pathways are either spatial specialization or temporal specialization. Ooh, space and time differences. Interesting. Here are some anatomical drawings then, showing the anatomical differences between C3 and C4 plants. So remember, in C4 plants, we can see the spatial separation in terms of the underlying anatomy. So notice in C3 plants, all photosynthesis is occurring in this upper palisade layer of cells, right? And so these then cells have to then deal with photorespiration. So in hot, dry environments, photorespiration starts to occur, right? But now check out the C4 leaf. Notice how different it looks in terms of the spatial separation. In C4 plants, the CO2 is first then intercepted by this PEP carboxylase, PEP, right? And then it's shunted or transported to the Calvin cycle or the carbon reactions then within the bundle sheath cells. So here are the bundle sheath cells. So we're going to be having the operation of rubisco or normal C3 photosynthesis here, but then we're going to have this outer layer, this outer onion layer of cells in kind of these outer palisade layer cells here, where we're having the operation of PEP carboxylase. Now remember, PEP carboxylase is a pretty cool enzyme because it is only a carboxylase. It's not an oxygenase. It does not interact with oxygen like rubisco does. All right. And in addition to the prevention then of oxygen fixation, there's also an increased resistance then to water flow then from the network because remember we have the xylem and the phloem right here, but because of the Krantz anatomy or this added spatial separation with this outer layer of cells, we have this increased resistance then of water then being transpired or evaporated then out. And because of which there's an added savings of water loss then within the plant. So this spatial separation then of C4 and photosynthesis results in this Krantz anatomy named after the German botanist who kind of actually discovered then this anatomy and coined then the term. You see these these circles or these rings, right? Where we have these inner bundle sheath surrounded then by these outer palisade layers, where we have the initial C4 photosynthesis and PEP carboxylase on the inside, we have Rubisco and the normal C3 photosynthesis occurring. So corn is a C4 photosynthetic plant. If you take a cross section then of corn leaves, you'll immediately see these bundle sheath cells, say that 5 times fast, surrounded then by these palisade then layers. Spatial separation then of C4 photosynthesis. So what are the central steps in C4 photosynthesis? So here then are the steps I would like you to memorize. And this is actually pretty easy. So the first step in C4 photosynthesis, and compare this with C3, is that the first step is not the carboxylation by Rubisco, but instead the carboxylation of PEP, okay, by PEP carboxylase. So we're going to be interacting with PEP, CO2, with the enzyme PEP carboxylase to produce this 4-carbon acid in these mesophyll cells. And these organic acids then, okay, this C4 acid is then going to be transported to the bundle sheath cells. Remember the Krantz anatomy? Okay. And then the third step is that these organic acids are then going to be carboxylated in the bundle sheath cells then by then producing then CO2. So we're going to be actively kind of concentrating CO2 within these bundle sheath cells. And then the fourth step is that CO2 is then going to be assimilated then by Rubisco and the normal kind of the dark reactions or the carbon reactions in the PCR cycle or the Calvin cycle. Okay. And then the fifth step is that these compounds are then going to return to the mesophyll cells to regenerate into more PEP. So here's my crappy drawing then of all of this, right? So this is to hopefully make it simple. Here, I should have chose green, I guess, but here, these blue boxes here, this is the epidermis then of the lower part of the leaf. Okay, so this is on the inside of the leaf, this is the outside of the leaf. Here's then an individual stomate. Okay, and so the stomates are open because here are the guard cells, and so they're open. And so the CO2 is then diffusing then on the inside of the leaf. Right? So the first then cell where the CO2 is going to diffuse in is these mesophyll cells. And so remember, this is where we have our different layers or spatial separation then of biochemistry. CO2 then is going to diffuse into these mesophyll cells. And so C4 plants then have this ingenious mechanisms to enhance the concentration or the partial pressure of CO2 at the site then of carboxylation then by Rubisco. So that this oxygenation reaction of Rubisco is basically limited and inhibited because we're artificially increasing the CO2 concentrations within the vascular bundle sheath cells. Okay, so note, this is kind of a textbook example of C4 photosynthesis. Turns out there's actually some variability. So I'm going to go through each of those central points that I showed you earlier. So here, the first step, okay, the second step, third step, fourth step, and the fifth step, right? So notice what happens first is that CO2 then is then going to be interacting with PEP carboxylase, with PEP, to produce this 4-carbon molecule, this 4-carbon acid. That 4-carbon acid then is going to be actively transported then into the vascular bundle sheath cells, okay? So this is our first step. PEP carboxylase, okay, is operating at CO2 concentrations about 100 micromoles per mole, okay? Low CO2 concentrations in general. Remember, it's hot and dry environments. That C4 then acid is then going to be actively then transported, okay, then within okay, these vascular bundle sheath cells. So notice then what we're also going to be what's also going to be occurring is that that CO2 then is going to be released because we have a decarboxylation event. Some biochemistry, a little bit complicated biochemistry, where we're releasing then that trapped CO2 in the C4 acid, releasing it into the vascular bundle sheath cell. That then creates a 3-carbon molecule that's then recycled then back into the mesophyll cells and kind of converted back into PEP again to complete the cycle. But notice what's happening here. We are now actively concentrating CO2 inside these vascular bundle sheath cells. Look at the kind of normal CO2 concentrations within the vascular bundle sheath cells, around 2,000 micromoles per mole, right? About an order of magnitude more CO2 kind of being concentrated here. So notice we have our normal kind of carbon reactions or Calvin cycle or PCR then cycle here. So we have normal C3 photosynthesis then occurring within the C4 plant, but now we've added this added layer of C4 photosynthesis. And the purpose here is to artificially increase CO2 concentrations in order to really limit, basically stop photorespiration occurring. So notice Rubisco here, it's all happy because it has this high concentration of CO2. Now, it turns out, if you take a little bit more detailed courses on this, that there are different flavors of C4 photosynthesis. And it turns out that these different types of C4 photosynthesis, I don't want you to memorize this, but there are flavors known as NADP malic enzyme or the NAD malic enzyme or the PEP carboxylase kinase. These then reflect differences in the type of enzyme involved in the decarboxylation of C4 compounds then to release CO2 then within the vascular bundle sheath cell. Okay. And I only want to introduce that to let you know that what you learn about photosynthesis normally in class is just a small taste of the variability in different photosynthetic pathways then. Okay. And it turns out that these differences in C4 photosynthesis are very important because different environments then have selected for then these different flavors in different way in terms of C4 plants. Okay. So what's the functional then result of C4 photosynthesis? Well, the functional result is that we are able to perform photosynthesis in environments then that would lead to low CO2 concentrations in the leaf because stomates then tend to be closed on really hot, dry days. And you can see that in the ACI curve. So here's the ACI curve, where again, we're plotting increases in CO2 concentrations, where Rubisco then lives, and we're looking at the rate of CO2 uptake from photosynthesis. But remember, this is the net rate of CO2, okay, from photosynthesis. So remember, net photosynthesis equals the gross minus the respiration. So notice the net photosynthesis increases in C3 photosynthesis, but we also have the gross photosynthesis, right? But also what's lost from respiration, right? And in particular, photorespiration. So here then is the difference between C3 and a C4 plant on an ACI curve. And if you do measure an ACI curve then in plants, an easy way to tell the difference between a C3 and a C4 plant is that the C4 plant will continue to do photosynthesis all the way down to CO2 levels extremely low. So notice that change then in the compensation point between C3 and C4 plants. That is reflecting this fundamental difference because C4 plants are able to scrounge for CO2 because of PEP carboxylase and then take that low concentration CO2 and then concentrate it in the bundle sheath cell so that they can still perform photosynthesis. So the compensation points then differ in C3 and C4 plants. Why is that? It's because C4 plants have PEP carboxylase that don't have the issue of photorespiration. And in the ACI curve then for the C3 and C4 plants, they differ. Why? Well, they differ because of the importance of PEP carboxylase and the fact that they have spatial separation of their biochemistry. All right, so remember, our net photosynthetic rate is the difference between the gross photosynthesis and your respiration. And in plants, C4 plants, they can limit then photorespiration, and because of which they can shift then that compensation point. All right, but C4 plants are not the only diverse kind of pathways of photosynthesis. There are CAM plants, this crassulacean acid metabolism plants. And you can tell CAM plants in general because they tend to be succulents. That is, these plants really thick leaves. Cactus, Cactaceae, wonderful CAM plants. Bromeliads, maybe you've seen bromeliads, or pineapples, right? A lot of epiphytes tend to be CAM plants. Now, what's interesting is that CAM plants, CAM trees tend to be very rare. There are a few, especially in the tropics, but CAM plants tend to be these succulent plants. It's actually interesting. Our earliest indication that these succulent plants are really interesting actually comes back to the Romans, who noticed then that there's something to do with maybe the metabolism of these succulent plants were different from other plants. And they realized that these plants differed in terms of kind of when they kind of operated differently during the day and the night. And they noticed this nocturnal pattern within plants then that we now know as CAM plants. And so the Romans actually noticed that certain plants, actually some edible plants, tended to taste more bitter in the morning than in the evening. I don't know if you've ever had kind of locally here in Tucson, nopales or nopalitos. You get, it's basically cactus, young cactus pads then that are chopped up and kind of fried up or kind of grilled or sauteed. Mix them with your eggs in the morning, a little bit of salsa on a corn tortilla. Oh my goodness, it's the best thing in the world. Well, anyways, if you get your nopalitos and you get them from a plant that was harvested in the morning versus harvested in the evening, you'll actually taste a difference between them. And so the Romans noticed this, that some of their succulent plants that they could eat were more bitter in the morning. So the rule of thumb was don't then eat the plants in the morning, eat them in the evening because they're less bitter. So that's kind of odd, right? Why would a plant taste bitter in the morning than at night? Okay, it didn't really make sense, but they had a rule of thumb and it worked. Well, it turns out that CAM photosynthesis results in increases in CO2 concentration, just like C4 photosynthesis, but CAM plants do it by temporal specialization instead of spatial specialization. So again, why increase CO2 concentration at the site of carboxylation? To limit photorespiration. So let's compare C4 and CAM photosynthesis. So here's a C4 plant like sorghum, corn, or sugar cane. These are C4 plants where we have this spatial separation, right? Where we're separating out our initial C4 photosynthesis, where we're creating then in these palisade then cells, creating this 4-carbon C4 acid, transporting it to the bundle sheath cells. We're decarboxylating that C4 acid, releasing the CO2 into the Calvin cycle or the carbon reactions, right? To do normal C3 photosynthesis, spatial separation. But within CAM plants, we basically do the same thing, but we do it in time. So at night, we perform effectively the equivalent of C4 photosynthesis. But then during the day, we switch over, close the stomates, and then we do the normal Calvin cycle. We decarboxylate then our stored CO2 and then perform normal C3 photosynthesis with Rubisco, again, during the day. Okay, so instead of separating out by the mesophyll and the bundle sheath, now we temporally specialize our biochemistry. Ingenious, but we don't have the Krantz anatomy, and we don't have the spatial separation with different cells. We separate it in terms of time between the night and during the day. Okay, my crappy drawing. Here's our typical CAM C4 photosynthesis, our typical cell within a CAM plant. So think of this as a cactus. At night, when temperatures are cool, relative humidities are higher, stomates then open on the cactus, okay, or a succulent plant, CO2 diffuses in. And then here we have the biochemistry at night. We have PEP, pep-carboxylate. We then make our C4, our 4-carbon malic acid, and we store that 4-carbon molecule, that 4-carbon acid, in the vacuole then of these cells. Stomates are open. Okay, daytime comes. Sun pops on, and then over the horizon starts to get really hot on these succulent plants, right? Cactus starts to warm up. Close the stomates to lower our rate of water loss, and so we kind of limit our water loss. And then during the day, notice the stomates are closed, but that's where we're now switching over then to our normal C3 photosynthesis, where we have our Calvin cycle or our PCR cycle or carbon reactions. We're taking then that C4 acid, okay, that 4-carbon molecule. We're decarboxylating it, releasing then CO2, okay, and then our normal kind of C3 kind of molecule. So notice we have all these different control points, right? Where we're now assimilating CO2 at night, okay, we're acidifying then the vacuole, and this occurs then the concentration in these cells where it's about 150 micromoles per mole. But notice, because we're dealing with pep carboxylase, that's not a big deal in those low CO2 concentrations. But during then the day, we're going to be taking then that malic acid out of the vacuole, and we're releasing then CO2 because we're decarboxylating then that C4 then acid, releasing our C3 molecule, which is known as CHO. And that CO2 level now within the cell is going to increase because our stomates are closed and we're decarboxylating all this malic acid. And now we can perform our normal C3 photosynthesis. So all of these products are then kind of recycled, okay? And so our C3 molecule, our 3-carbon molecule, is going to build up then during the day so that as to be present and in high concentration then at night, okay? And notice we're now artificially increasing the CO2 concentration inside the plant to, again, over 4,000 micromoles then per mole, which is really crazy. So now Rubisco can operate at these really high CO2 concentrations, and photorespiration is effectively eliminated because relative concentrations of CO2 are much higher than concentrations of oxygen. Okay. So we can see this temporal specialization if we plot out the relative concentration then of all of our different players, such as, for example, our malic acid, our C4 acid. So notice at night, when the stomates are open then within the cactus, CO2 then is diffusing then into the cactus itself. Our pep carboxylase is then interacting then with the CO2, creating then our C4 malic acid. The acids then build up. The plant is now tasting really bitter, okay? So by early morning, we have our highest concentration of malic acid. But then as soon as the sun comes up, the stomates close, right? Our biochemistry fixes, kind of switches over. We start decarboxylating all of that malic acid to release all the CO2, right? And we start to get CO2 fixation then by Rubisco during the day with the stomates closed, okay? So CO2 is being fixed by both pep carboxylase and Rubisco, but it happens at different times of day. Pep carboxylase at night, Rubisco during the day when the stomates are closed. Ingenious. Now, because of which, how C4, CAM, and C3 plants then use light, water, and nitrogen actually varies because of spatial and temporal specialization. Okay. So the light use efficiency actually differs quite dramatically between C3 and C4 plants. So if you plot our light intensity, or photosynthetic photon flux density, versus the net photosynthetic rate, notice in C3 plants, photosynthesis is kind of maxed out at low levels because of photorespiration. Because high light levels mean the plant then is heating up, stomates are closing, more photorespiration. C4 plants don't have that issue. They can have higher photosynthetic rates at higher light levels because they don't have to deal with photorespiration. Okay. Now notice if you plot a C3 plant in the absence of photorespiration, it falls right in line with what a C4 plant does. And that's basically the specific adaptations of C4 photosynthesis, is to get rid of the effect of photorespiration. Ingenious. Okay. But also, if you look then at different kind of temperatures and look at the light use efficiency, how efficient then these different plants then use light, it's only at these higher temperatures where C4 plants are actually much more efficient at using light. Whereas C3 plants, when it's colder, cooler out, are actually much more efficient in terms of photosynthesis. Okay. So with climate change, global warming, we're warming then the planet. And because of which we are providing a natural selective environment for differences in photosynthetic pathways on the planet. C4 plants do much better at higher temperatures. Okay, so recall water use efficiency. All right, so water use efficiency is our photosynthetic rate divided then by our transpiration rate. Okay, so water use efficiency is photosynthesis divided then by water use or transpiration rates. So notice photosynthetic rate from Fick's law is just the difference between internal and external concentrations of CO2. Same applies to water use, Fick's law, difference between internal and ambient water concentrations. The ratio then between photosynthetic rate and transpiration rate is the water use efficiency. So again, C4 and CAM photosynthesis then changes then the demand function of photosynthetic activity, and also then changes plants' water use. So if you're a crop breeder and you want then to improve the efficiency of crops growing in a hotter, drier environment, water use efficiency is one kind of critical trait then that you try to select on, because we want crops then to be as productive for a lower amount of water than being used, because water is increasingly in shorter demand. Drought environments, water is hard to find. So in C4 and CAM photosynthesis, they can lead to reductions in E, or your transpiration rates. So remember, in CAM plants, they actually close stomates then during the day when the water then loss is the greatest. Whereas in C4 plants, you're able then to kind of conduct photosynthesis at a higher efficiency when stomates are increasingly more closed. Okay, and because of which C4 and CAM plants have a higher water use efficiency. That is, they're more productive for a given amount of water than that they use. And so because of which this results in kind of changes in stomatal conductance, temporal dynamics of stomatal behavior. And the consequence of all of this is that in C3 plants, for 1 gram of biomass then that's produced, you need 650 to 800 grams of water that's lost or transpired. Wow, that's amazing. So to grow 1 gram of C3 biomass, you need 650 grams of water transpired. That's 2 orders of magnitude more amounts of water relative to how much you're going to gain. Compare that to C4 plants. For C4 plants, for 1 gram of biomass produced, they're much more efficient. They're better users of water. So instead, they only need 250 to 350 grams of water transpired. Okay, about half then is less. CAM plants, on the other hand, for 1 gram of biomass, they only need 100 or 200 grams of water transpired. And because of which, this is why CAM plants dominate desert environments. Cactaceae cactus, they're CAM plants. They're much more efficient at using water. Okay, so our water use efficiency then decreases as we move from C3 to CAM. All right, there's other savings as well and other efficiencies. Nitrogen use efficiency. So remember, Rubisco in C3 plants represents about 30% of the total leaf nitrogen, sometimes up to 50% of leaf nitrogen if you include all the photosynthetic enzymes. And C4 plants in general have 3 to 6 times less rubisco than C3 plants because they're more efficient, right? And because of which they actually need less nitrogen in their leaves, okay? So the nitrogen use efficiency then is actually much higher in C4 plants. CAM, on the other hand, is a little bit more variable in its nitrogen requirements, but in general, C4 plants tend to be better at using than nitrogen. They're more efficient at using their nitrogen. Okay, so what were the evolutionary pressures then leading then to the evolution of C4 and CAM? Well, in general, again, this is selection for increased water use efficiency and or possibly nitrogen use efficiency as well as the main selective pressures, okay? Because C4 plants, especially in hotter and drier environments, more efficient at using not only water, but in general, C4 photosynthesis is also more efficient at using nitrogen. Now, what's really interesting is that it looks as if C4 photosynthesis has likely evolved independently many times. It just didn't evolve once. We know now that C4 photosynthesis has evolved independently many times. And it first evolved around 12 million years ago, okay? Relatively not that long. So remember, around this time, this was also the period then when the Earth then started to not only dry, but also warm kind of considerably. Now, we only find C4 photosynthesis within then the angiosperms, mainly within the monocots, but also in several eudicot families. And in general, we tend not to see C4 photosynthesis within trees. So within grasses, herbaceous individuals, shrubs, and so on. And during the evolution of C3 photosynthesis, atmospheric concentrations of CO2 were much greater. So around 12 million years ago, not only did the planet start to not only warm and get a little bit drier, but CO2 levels started to reach really low concentrations. So with the success then of the land plants, CO2 concentrations in the atmosphere actually started to decrease. And around 12 million years ago, CO2 levels were actually pretty low in the atmosphere. But at the same time, remember, oxygen levels have increased through time. And this is kind of like this perfect storm that around 12 million years ago, the rise then in this, what could be a flaw or this Achilles heel in C3 photosynthesis became increasingly more clear. Photorespiration became much more prominent. And so these are all conditions that favor the increase in photorespiration. And this also led then to conditions that favored then for selection to increase in some way, if there was variability, some traits then that led to these CO2 concentration specific adaptations. So the factors favoring C4 evolution, low CO2 concentrations. So this ratio between CO2 concentrations and oxygen, the relative ratio, started to change around 40 million years ago, and definitely then as we then headed into increasingly into the Pleistocene. In general, this led to increased photorespiration then within C3 plants. And secondarily, we have higher day temperatures and more dry or limited water environments. So water use efficiency is kind of regarded as the main selective trait favoring the evolution of C4 photosynthesis in certain environments, especially. So we have then selection in certain environments favoring C4 over C3 photosynthesis. Again, hot, dry conditions, low CO2 relative to the concentration of oxygen. And in general, we can kind of predict where on the planet this switch between C3 and C4 plants are going to be favored based on this biochemistry and when photorespiration tends to increase. So if we plot the daytime growing season temperature, increases in temperature, cold into warm versus the atmospheric CO2 concentration, low CO2 concentrations, think going back 40 million years ago, kind of into the Pleistocene, okay? Atmospheric concentrations were really low. But as we start increasing CO2 concentrations, okay, that also switches then this flipping point, okay, between environments where C4 is favored versus environments where C3 are favored. Notice C3 photosynthesis is favored under cooler conditions and higher atmospheric concentrations, whereas C4 photosynthesis is favored then under increasingly warmer conditions, okay? But then also C4 photosynthesis is favored then under lower CO2 concentrations. This all has to do with this relative flip when photorespiration becomes increasingly, having an increasingly more negative impact on C3 photosynthesis. So that's pretty cool. So notice we are changing the planet. We're putting more CO2 into the atmosphere. And as a result, we're warming then the planet up. But depending then on the relative impact in terms of both CO2 and temperature, that will influence what photosynthetic pathway is favored in future worlds. Okay, so other hypotheses. There's a few other hypotheses for kind of the evolution of C4 photosynthesis. One is that herbivory may have been important. That is, C4 plants, because they're less nutritious than C3 plants, then they kind of escape herbivory. And maybe actually this could have been another kind of factor leading then to the kind of the increase in C4 plants, right? So C4 herbivores actually tend to be really specialized in a way because the nutrient concentrations in C4 plants are really low. And so it could be that C4 photosynthesis could lead to an escape from herbivory as well. Right? And so what's really interesting is we start then to look at kind of details of the evolution of C4 plants. It actually turns out that there's a lot of variability. And in many taxa, it's not really clear whether or not the plant is C3 or C4 because there are these interesting evolutionary transitions possibly on these additional kind of intermediaries between the two. And it turns out that these C3, C4 intermediaries are really important in understanding then the evolution of C4 photosynthesis. And so taxa then that are intermediary, kind of in between C3 and C4, appear to have evolved multiple times. And you can see these in multiple families across the angiosperms. And in particular, I kind of want to highlight kind of one group of plants. This is a group of plants studied by Russ Monson. He's actually here at the University of Arizona. This is Flavaria. And these are kind of interesting kind of shrubby composites or these sunflowers, right? And they grow in these different environments. And if you look at the phylogeny then of Flavaria, what you actually find is that several of these are different species then of Flavaria. And many of these species are C4 Flavaria, but then there are some Flavaria that are C3, which is interesting. So we get nice Krantz anatomy and this kind of spatial separation then of their C4 photosynthesis. But notice we have other plants that are these intermediaries between C3 and C4. They kind of do both, right? And so it kind of looks as if we map this on a phylogeny, then that kind of initially the common ancestor was a C3 plant, but then we get these independent kind of switches to C3 and C4, and then the evolution of pure C4 photosynthesis kind of independently. And this is in this one little clade within this one genus of plant within Flavaria, where we can see the independent evolution of these intermediaries, lineages then that have both C3 and C4, and then independent evolution of C4 photosynthesis just within this one little clade. And so it turns out C4 photosynthesis has evolved multiple times. Seemingly, it's kind of easy for evolution to select and kind of evolve C4 photosynthesis. CAM evolution is a little bit more weird. CAM actually evolved from C3 plants before the evolution of C4 plants. And actually, we see evidence of CAM plants evolving, gosh, way back in the tertiary, 20 to 30 million years ago, actually an aquatic species of all things. And it's actually found in 2 times as many families than C4 plants. So CAM photosynthesis is actually much more prevalent across the land plants. And it's actually pretty diverse. We see CAM photosynthesis within lycophytes. Gosh, remember the lycophytes, the zostrophyllophytes, within pteridophytes, ferns, within the cnidophytes, within the gymnosperms, and also within the angiosperms. Oh my goodness. CAM photosynthesis occurs sprinkled throughout the land plant phylogeny from lycophytes, pteridophytes, cnidophytes, and the angiosperms. Remember, C4 plants, we only see that within the angiosperms, in particular, especially within Poaceae, within grasses and the monocots. So we tend to see CAM evolution reflecting the strong selection to maximize carbon assimilation and selection to overcome CO2 limitations during the day. Okay, probably the result of having closed stomates in increasingly drier environments. And in general, 6% of the terrestrial and aquatic flora is actually CAM photosynthesis. And unlike C4 photosynthesis, some CAM plants can actually grow in really deep shade. And there's even CAM plants that grow at very high elevations. And the more we look, we actually find CAM photosynthesis in many different diverse environments. And it's usually associated with succulents, the evolution of succulents. So if you're familiar with cactus, remember cactus plants, ice plants, stone crops, euphorbs, bromeliads, orchids, okay? About 50% of orchids are actually CAM. A lot of tropical forest epiphytes, they grow in the dry kind of dry tree branches up in the canopy. Many of those epiphytes tend to be succulent. So any sort of kind of succulent plant then tends to be associated with CAM photosynthesis. You even see CAM photosynthesis in aquatic vascular plants, where it's presumably it enhances the ability then to kind of increase the concentration of CO2 because in aquatic environments, it turns out CO2 availability can be rate limiting for photosynthesis. Okay, so in general, we see CAM photosynthesis in kind of environments where kind of in general, we see the evolution of succulents. And in general, the presence of CAM declines when nighttime temperatures actually tend to get really, really high. Okay, so our deserts here in the Sonoran deserts, these are actually kind of cool deserts in general. We tend to cool down at night and we tend to cool down, especially during the winter. Okay, so some crazy CAM plants. Remember Welwitschia? Remember our crazy Welwitschia and the cnidophytes? A lot of different orchids, houseplant Sansevieria or mother-in-law's tongue, pineapples, cool tropical trees. Actually, these clusia trees, they're kind of these rare CAM plants, agaves, cactus, all of these more succulent plants, all of these are CAM. And so what I'm kind of hinting at, the more we kind of study differences in photosynthesis, kind of the simplicity that we've learned about C3 photosynthesis, in many ways, the more and the more we kind of study diversity of photosynthetic pathways, we actually find that plants are remarkable and quite variable in their photosynthetic strategies. And to just kind of illustrate this diversity of photosynthetic strategies, I'm going to highlight this one plant, ice plant. If you've ever been to California, this is a plant that tends to grow around the sand dunes of a lot of Southern California beaches and even kind of beaches kind of further north. This is a nice house plant too, that's originally from South Africa, but has been introduced kind of all over the place. And so you tend to find these in these really arid, semi-arid places of Southern Africa, and now increasingly throughout the world. You tend to see it in, you can also kind of plant it in around Tucson. Succulent plant, right? So if it's succulent, you tend to think, all right, this is CAM photosynthesis. And so what's really crazy about ice plant is that it kind of displays these amazing diversity of photosynthetic responses depending on the time of year. And in general, it actually starts to act like a C3 plant and does C3 photosynthesis at certain times of the day, okay? And so if you actually notice here, what this ice plant tends to do is that if you then kind of look throughout the year, okay, and we're looking at the leaf water content. So during the rainy season, okay? As we move kind of through the rainy season, the leaf water content is really high. But as the year then passes on and we enter into the dry season, the leaf water content starts to drop. And as the environment becomes more stressful, this one plant will perform C3 photosynthesis normally. So all the biochemistry we've learned. But then as soon as the environment starts to get a little stressful, it switches over and it kind of starts upregulating the CAM photosynthesis, right? And if you notice the leaf water content as it starts decreasing, it starts to switch over its biochemistry and switches then to CAM. So notice if you look at the malate concentration, okay, within the cells, then remember malate, this is the C4 then kind of acid then that's produced and stored then within the vacuoles of CAM plants. Notice in the dry season, it starts to become more acidic, okay, as we then look across then the time. And what's interesting, if you look at the total photosynthetic rate, for the most part, there's a little bit of ups and downs, but for the most part, photosynthesis is maintained kind of constant through the year. So this switching over then between C3 and CAM within this one plant is this ingenious kind of way that natural selection then has dealt with a highly variable seasonality. As we go from wet seasons to then dry season, really hot, arid, desert-like conditions, then certain plants then have the ability to kind of bring out these additional photosynthetic tools and then switch between between C3 and CAM in this way. If you've ever been to the Central Valley of California, especially coming out of the wintertime, you'll see these vernal pools. These are amazing kind of locations to do botany anyways. And so during the wintertime, California normally gets a lot of in the Central Valley. Excuse me, my microphone is, okay, I think I'm back. My microphone shut off, but I'm now back. So anyways, these vernal pools then fill up during the winter time. And as then the vernal pools dry, as the hot, dry California summer kind of approaches, no rain then falls. But as these pools then dry up, you'll see then all these different species of plants kind of taking advantage then of this water. You get these wonderful blooming events. Incredible diversity associated with these vernal pools in Central California. And so the environment changes from very wet to very dry. And the California summer, especially in the Central Valley, can be very hot and very dry, almost desert-like. And so if you look at a few of the plants growing there, in particular, this one lycophyte, oh my gosh, it's a lycopod, right? Really cool little plants. Anyways, so physiologists have studied then how these plants respond to this highly variable environment. So this quillwort, this lycopod, is CAM when it's actually submerged in these vernal pools. And then once then the water levels then drops, it switches to C3 photosynthesis when it's not submerged. And so these different species actually, there's these different species then of these quillworts all growing in these vernal pools. So one of them switches from CAM to C3, okay, whether or not it's submerged or not. But these other ones then use these kind of unique kind of channels, these specific adaptations, and these pores of these channels in the roots to enhance CO2 uptake then from these submerged then soils. Also within these vernal pools, you'll see other species, such as these aquatic grasses, that uses C4 photosynthesis when they're submerged. But then there are other aquatic plants that uses C4 photosynthesis when they're submerged, but then shift to C3 photosynthesis when they're exposed then to the atmosphere. Now, this is all in this one little local environment that when we start to look in much detail, we actually see that C3, C4, CAM can be employed kind of differently in these different plants, all within this one localized plant community. And these are different strategies then to deal with this highly variable environment. Differences in water supply and the ability then to uptake CO2 in either aquatic or increasingly drier, hot environments. And all these strategies likely evolved independently then from each other. And so what we're learning is that this photosynthetic diversity is actually quite amazing. And it's reflecting differences in selection for dealing with these highly variable environments, especially in these variable environments that are hotter and drier. Okay. Now we can see the geographic distribution of this photosynthetic diversity as we look across the landscape. And so this is associated with changes in the relative importance of these different selective pressures on water use efficiency and nutrient use efficiency, having to do with differences in temperature and variability in water supply. And so this is kind of a crude little map here, where we're looking at the percentage of grass taxa that are C4 plants, and the percentage then of taxa, actually eudicots that are C4 plants. So this is grasses on the left, and then eudicot plants then on the right. So if you notice, as we move kind of from these hotter, sometimes drier environments, so really hot environments, to increasingly kind of cooler and more wet environments. So it's wetter on the East Coast, wetter in the Pacific Northwest. Notice how we go from a high percentage then of grasses that are C4 here in the desert Southwest to a very low percentage then that are C4 in these colder, wetter environments. Same thing for eudicots, okay? And so we see increasing percentages then of eudicots then that are C4 plants, especially in the desert Southwest, hot, dry environments. But then as we move into these wetter, colder environments, that percentage drops down in some locations to basically nothing. So if you're studying Arctic and alpine physiology and photosynthetic diversity, basically most everything is C3, but if you move into these drier, hotter environments or highly seasonal environments, we see increases in C4 photosynthesis, as well as CAM as well. Okay, but remember I told you there are these different flavors then of C4 photosynthesis. And a really good predictor of the percent of C4 photosynthesis in the different species then that you'll see in the local flora is actually the average January minimum temperature. So if it's really cold out, you get a really low percentage of C4 species. As you get increasingly warmer winters in the January time, a very high percentage of the grass flora is C4. But notice with changes then in mean annual rainfall, these different flavors, remember these were the different flavors of this enzyme involved in decarboxylation, right? These different flavors of C4 then are going to vary with difference then in mean annual rainfall. So differences in temperature, differences in rainfall is then going to influence then the types then of photosynthesis then that we see occurring. Okay, so that's the end of our little lecture here on photosynthetic pathway diversity. So you should be able to outline some of these differences, fundamental differences between C3, C4, and CAM photosynthesis. And you should outline not only the similarities, but also the differences then between them. You should be able to tell us some of the unique selective pressures that led to the evolution of C4 and CAM pathways. But also you should be able to have an informed discussion about why all plants are not C4 or CAM photosynthesis. Okay, so thank you very much. I'm going to end it there, and I'll see you at the next lecture.